In her first presentation, Emily P. introduced us to the concept of genetic modification. Since it was quite a while ago, here's a quick reminder about the process of genetic engineering.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, EcoR1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
The DNA fragments produced in large quantities by the bacteria can then be inserted into organisms using other bacteria, viruses, or other methods which we will discuss later. These organisms are now considered genetically modified. Since proteins control all life as we know it, any organism can produce the proteins coded for by any DNA. Any organism can express any gene. We are going to talk about the controversy surrounding genetic modification. The first of which is genetically modified foods.
Genetic engineering allows us to quickly and accurately enhance desirable traits in genetically modified foods. For example, if we want a tomato that grows better in dry climates, we can take a gene from, say, a cactus, and implant it into a tomato and clone many plants that have the same drought resistance gene. This works for any particular trait. Simply find an organism that has the trait you want, isolate the gene or genes that codes for the trait, replicate the traits as described earlier, and insert them into a plant seed. These genes can also be patented, so only one company can sell plants with that gene. Because of this, one company, Monsanto, has 90% of the GM seed market. Sounds great, right? But not so fast. The majority of GM foods are Monsanto's Roundup Ready plants. These plants have an engineered resistance to Monsanto's Roundup herbicide. The process they use is relatively similar to the process used for all genetic modification.
Genetic engineering is really a radical revolution in food production. It's really a cell invasion technology. You know, people have heard they're taking a flounder gene and putting it in tomato so the tomato can last in cold temperatures. But people don't ask, how does that flounder gene get in that tomato? How does it get in there? And what really happens is, the only way you can do it is to invade the cell of the tomato and deposit the flounder gene. Well, what's good at invading cells? Bacteria and viruses.
After 12 years of searching, Monsanto found a soil bacteria that is naturally immune to Roundup herbicide. Their goal was to genetically engineer DNA from these bacteria into various plants. They cut out a sequence of DNA that is resistant to Roundup. But if this DNA sequence alone is inserted into a corn plant, it will have no effect. So the next step involves E. coli bacteria. Gaps are created in the E. coli DNA, and when the two test tubes are mixed together, some of the E. coli DNA recombines with the Roundup-resistant bacteria. Then the technicians smuggle the engineered DNA into the cells of the corn plant they want to modify. Cells will naturally reject foreign DNA, so they developed a method using soil bacteria that causes tumors in plants. They use this bacteria to ferry the engineered DNA into the plant's nucleus. There are also 2 other methods used to get the engineered DNA through the cell wall. One uses a stream of electricity to create tiny holes in the plant cells so they become vulnerable to infiltration by foreign DNA. Another is the gene gun, which blasts particles of gold coated with engineered DNA into the plant cells. Each of these 3 methods needs a promoter gene that turns on the desired characteristics. The promoter gene is often extracted from the cauliflower mosaic virus.
Another major GM food topic is Bt corn. Bt corn contains genes from Bacillus thuringiensis bacteria that produce crystal proteins which are lethal to insect larvae. It's used in 14 countries. There are 600 different strains of bacteria, so there are many toxins that each affect a specific organism, such as flies or beetles.
As with any controversial issue, the public is split between 2 views. But the real question is, what's the truth?
Nobody knows. Monsanto managed to get U.S. government regulations passed that essentially give them free rein to introduce whatever genetically modified foods they want. This means no government testing of GM foods is occurring or has occurred, and private research into GM foods is also very limited. Most private research is funded by companies or the government, and neither of these sources show any inclination to prove or debunk the myths or facts surrounding GM foods. Public opinion is very much split on the pros and cons of GM foods. We will address each of these later. Those in favor of GM foods make many claims about the benefits. Proponents claim GM foods will increase the total yield of farms. However, some studies have shown that GM foods actually reduce the total yield of a field, despite popular belief. Most likely this is because farmers don't use crop cycling techniques because they don't have to do so anymore in order to avoid pests. Monsanto trumpets the fact that Roundup-ready plants let farmers use Roundup exclusively to count weeds in their fields. Doing so can increase crop productivity. However, the ability to use a single herbicide in large quantities leads to the general deterioration of farming practices, which has the end result of lowering the total yield. Companies claim they can increase the nutritional value of foods. However, none of them have succeeded in doing so, leading to the conclusion that it's not possible or in their best interest to do so. Proponents also claim that GM foods are a way to feed the rapidly growing world population. However, all of these claims have yet to be proven, and when combined with the fact that creating a new GM food is a long and expensive process, mean there is no solid proof that they actually are a way to feed the future.
Those opposed to GM foods also make many claims about the detriments. Some plants are engineered to produce their own endogenous pesticides. This means that anybody who eats the plant is also consuming the pesticide. This is especially concerning when you realize that Monsanto, the main purveyor of GM foods, has a record of not reporting harmful effects of their products. So these pesticides could be very harmful to humans. However, this is unlikely, although untested, that these chemicals survive the harvesting and preparation processes. Many opponents argue that GM foods cause cancer. Monsanto says they know their products don't cause cancer. But as far as we know, nobody knows everything that can cause cancer. But Monsanto is claiming that they know the company doesn't use any of these unknown carcinogens. Sound funny to anyone else? Either they know everything that causes cancer and they're withholding it from the public and other scientists, or they're lying. Either prospect isn't good. Another claim of opponents to GM food is that they cause precocious or early onset female puberty. As with cancer and pretty much anything else, there hasn't been any research into this. However, as with cancer, Monsanto's claims are highly suspicious. Although Monsanto passes blame to improve nutrition, the University of Michigan website on precocious puberty or early onset puberty lists that many possible causes, but improved nutrition is not one of them. The Mayo Clinic document on precocious puberty doesn't mention nutrition either. One has to wonder where Monsanto is getting its information from. Skeptics of GM foods also claim that they are the cause of increase in allergies seen across the developed world. As we discussed in class, autoimmune disorders are increasing everywhere. Allergies are the main autoimmune disorder, and so far studies have not clearly linked this increase to GM foods. They probably never will, and this claim is likely impossible to prove or disprove because so many factors contribute to the development of allergies.
These aren't the only issues with GM foods. There is a risk of unintentional patent infringement because some of these GM crops invade other fields and eventually will turn all the crops into the GM proprietary food. There are currently court cases between Monsanto and farmers about Monsanto's proprietary seeds. The farmers never purchased Monsanto's seeds, but their neighbors did. The GM crops then basically invaded and took over the non-GM farmers' fields. Monsanto is claiming patent infringement and is suing the farmer, even though, as far as anyone can tell, he had no ways of preventing the invasion. Additionally, most of the genetically incorporated pesticides kill both good and bad insects. Some scientists have hypothesized that this is one reason for colony collapse disorder among honeybees. Additionally, the practice of genetic modification leads to plants that are genetically identical. Thus, one new disease would wipe out an entire species. The final straw is that a recent studies found that microRNA fragments from GM foods can and are absorbed by mice through ingestion. They interfere with the removal of LDL from the mice's blood. While this may not alone be a case to stop GM food production, it definitely is a cause for increased regulation and testing, which Monsanto openly claims is useless and unnecessary. There is much more controversy about this topic, chief among which are Monsanto's questionable claims and the incredibly lax, almost non-existent, regulation of GM foods in the U.S.
In the United States, regulation is nearly non-existent. FDA standards are the same for GM foods as they are for foods produced by natural breeding. What regulation that is in place is based upon the false idea of that GM foods are substantial equivalents, meaning that new genes will be no different from what is normally in plants eaten by people. A former FDA official admits that the meaning behind this sentiment was solely based on DNA. Humans consume DNA, and the new DNA is the same as any DNA. This is not at all based upon what the DNA is actually doing in the plant or how it has altered it. Although there seems to be no harmful effects of labeling, the U.S. refuses, probably at the behest of Monsanto, to require GM food labeling, essentially keeping the people in the dark about what exactly they're eating. In Europe, there is very strong force against GMOs. All GM foods are required to be labeled. The attitude in Europe is along the lines of, we don't know what will happen, so we should be careful, rather than the US's approach of, show the consequences and maybe we will stop the use after they've already happened.
Our second topic is genetically modified animals. We've all seen the pictures of glowing rats and pigs with glowing noses.
But what is the point of genetically modified animals? There are many potentials for this technology. Animals are currently being engineered to produce drugs for both animal and human use, decrease pollutants in manure, and serve as cell factories for human tissues. Scientists are also manipulating the genes of animals in order for them to produce high-value organic materials, antimicrobials to prevent food poisoning, and to produce healthier and more efficient food animals, such as the cow and fish pictured here. These practices, on the surface, seem wholly beneficial to society. So why is this so controversial? It has been reported that many animals designed this way are born with deformities and die very young. Additionally, if infected animal tissues replace human transplants, the human could contract whatever disease the donor animal had.
This presents a wide range of problems on the surface, although most animal diseases are not communicable to humans. With appropriate screening, this could almost certainly be prevented.
Critics of genetic modification insist that the methods are unnatural, and many deem the practice akin to playing God. This is the main question with animal genetic modification. Is the practice ethical on the whole? The argument against GM animals is perpetuated due to the risk of misuse of the technology. Taken to the extreme, it is possible that one could genetically modify animals to do their dirty work for them. Those in favor counter with just because genetic modification isn't allowed, it won't stop individuals from doing it. Improving the technology and implementing regulation and oversight is a better method to prevent misuse then shoving it under the rug and turning a blind eye to the situation. The method is also critiqued because it disrupts the natural genetic material. Mother Nature spent millions, even billions of years perfecting DNA, and we've only just discovered it in the past century. Do we really know what we're doing? The answer is no, we don't. We have a pretty good idea and grasp on the concept, but if we knew what we didn't know, we wouldn't not know it, right? Genetics is a very new field. Only recently has the field of epigenetics come into light. This is the study of how environmental factors change gene expression. This area of study is fascinating, as well as incredibly complex.
Here's a clip from Jurassic Park that shows what happens when scientists don't really know what they're doing.
Could be in DNA.
What's that?
Well, on the tour, the film said they used frog DNA to fill in the gene sequence gaps. They mutated the dinosaur genetic code and blended it with that of frogs. Now, some West African frogs have been known to spontaneously change sex from male to female in a single-sex environment.
The movie scientists didn't know the DNA they used would allow spontaneous sex changes, which enabled the dinosaurs to breed.
The controversies surrounding GM animals are difficult to support or refute because, as with GM foods, research is scarce and rarely peer-reviewed because it's funded by companies with vested interests in GM animals. Additionally, the procedures of these studies are not reviewed by the FDA, which bases its decisions upon the studies. Thus, the regulations and approvals are based upon what could very easily be fraudulent science. The answer to this is to have more well-constructed research. The difficulty with this is funding. As mentioned during the plants section of the presentation, much, even most, research funding comes from companies or the government. The way to go about this is to figure out how to slant the research proposals to portray a benefit to some company. Additionally, animals are genetically altered to have an advantage over whatever is the natural state of that animal. Thus, if they escape, which is probable, they might outcompete the natural type, and the natural type could be lost forever. We could take a Jurassic Park-esque approach to this and alter the animals so they require a substance that can't get in nature. In the books and movies, the dinosaurs were engineered to be incapable of producing lysine, the molecule shown on the right. Thus, if they escaped, they would soon die of lysine deficiency. Genetic engineering technologies could replicate this scenario in GM animals to prevent the introduction of engineered species into the natural environment. Another concern with GM animals is if we were to make an animal resistant to a very strong antibiotic, so we can treat them with previously toxic levels, the pathogen could acquire immunity and then run rampant because we don't have a way to control it, similar to the situation we now face with MRSA. This concern is something that regulation would have to take care of. It is potentially foreseeable that industrial complexes will use genetic modification as a way to compensate for incredibly sloppy techniques, such as not trying to avoid fecal contamination because the animals can resist the bacteria. Regulation should be implemented that prevents this slackening of standards as a result of GM animals. Just because the animals can resist the disease doesn't mean we should expose them for no reason. The basis for all of these claims is quite simple.
This capacity of bacteria and viruses to invade mammals in different ways is what really has a lot of people edgy about biotechnology, because that's really what the tools are all about. In order to move genetic material from one organism to another that don't normally cross, you've got to sort of behave like bacteria and viruses and invade into the cells and become established, just like a virus must become established.
The third and final field of genetic modification is colloquially known as designer babies. So what capabilities do we have? The most common and best refined technique is pre-implantation genetic diagnosis, or PGD, a method used for gender selection and also used to select physical traits. Its original purpose was to screen for disease. A similar technology is germline therapy, which is a method of replacing genes that would cause disease with normal genes in embryos. All of the other things we talked about earlier apply to this too, such as taking non-human genes and putting them into humans. The following video gives an example of this ability.
The very first gene that was added to an embryo was a gene coding for growth hormone. It's a gene that causes an animal to grow larger. And a growth hormone gene from a rat was placed into a mouse embryo. And when the mouse was born and it grew up, it grew to twice the size of a normal mouse.
The benefits of these methods are much simpler and more important than just increasing the size of a human. Eggs may be tested in vitro for a specific disease, and unaffected eggs may then be selected for fertilization, thus guaranteeing offspring that are disease-free. Through these processes, eventually we might be able to eliminate certain genetic diseases. Additionally, it would allow us to choose good traits, what we want the rest of humanity to display. But that brings us right to the controversy surrounding designer babies.
Let's jump right in. What are these controversies?
Because it would be very expensive to perform these tests at first, the socioeconomic difference in humans could become a genetic difference. Only the rich could buy disease-free super babies. Some people believe that because of the price factor, in the future there will be two breeds of humans.
The whole topic of enhancement, I think, causes people pause. It causes me pause. Suppose we develop, by our understanding of how the genome works and therefore how the body works, an approach that would improve memory. What's wrong with that? Or an approach that would make all of us able to stay slender, even if we were being fairly careless about our diets. What's wrong with that? Well, it does raise the question about who decides what's an improvement. And is that improvement something which is going to be available to all, or will it be another example of separating between people who have resources and people who don't?
The key question is, what happens to society if everyone is clamoring to have their status and their capabilities boosted? And in a worst case scenario, what happens if society itself begins to fracture? On one hand, into a race of super beings, and on the other hand, the rest of us.
Modifications are hereditary, meaning that they are transferred to offspring. In other words, these modifications are changing the very genome of human beings, which could have dire or amazing consequences.
If we could pass down these genetically enhanced genes, we could evolve in a different way. This is a serious issue to be waking up to now in 2007, and I'm concerned that this advance in biotechnology that makes it conceivable to modify the human genome, and we see this, that these procedures are possible, are happening, that we're not thinking actively enough about what the impact will be 15, 20, 30, 100 years from now, and we can't afford not to think about it and not to be prepared.
We're at an inflection point in history. For the first time in hundreds of thousands of years, our technologies are not so much aimed outward at modifying our environment in the fashion of fire or clothes or cities, agriculture, space travel. Increasingly, these technologies are aimed inward at modifying our minds, our memories, our metabolisms, our personalities, and our kids.
IUD was originally intended to be used for the prevention of disease, but the procedure could eventually be used to select for intelligence or specific skills, creating unfair competition and selection between individuals. As the earlier video mentioned, this also brings forth the question of what's an improvement.
Tell me, who exactly is to decide what a desirable trait is? It gives an example in your textbook of people who wish to eliminate aggressiveness. But couldn't it be argued that aggressiveness is essential to some aspects of life? An attempt to stamp out some traits implies that there are some human beings inferior to others, and this would create quite an uproar. Of course, some desirable trait that scientists wish to engineer into humans could include strength and intelligence, which seems like an advantage. But in the long run, couldn't the same methods be used to work in the opposite way? While it may sound absurd and futuristic, just like they could engineer people to be so much smarter, what's to stop scientists from modifying humans to only follow orders and get things done? I mean, doesn't the world need those people too?
Animal testing would be required for any gene to be used in humans. Perhaps the most likely animal candidate for this is chimpanzees. But what happens if we try to genetically alter humans to be incredibly intelligent or really strong and test these in chimps? we would end up with really smart apes, or really strong apes. Or, more worryingly, really strong and really smart apes. Planet of the Apes, anyone?
Because chimpanzees share 99% of their DNA with human beings, they're essentially identical to us. We can test the safety of the human artificial chromosome by placing it into a chimpanzee embryo, many chimpanzee embryos, and making sure that the chimpanzee babies that are born don't have any birth defects. If we're trying to increase the intelligence with this artificial chromosome, we actually might end up with a chimpanzee that has human or greater than human intelligence.
So what are the cold hard facts? PGD can screen for diseases and traits, but is currently only used for the former. PGD is regulated by both the CDC and the FDA. The only method normally in practice is PGD, which means children cannot express any genes that aren't in the parents' genomes. In other words, embryos can be created and screened for diseases and traits, but currently the only option is to choose between them, not to alter them. The technology to modify embryonic genes on a large scale are probably years away from implementation. As with any new technology, initially this will be very expensive. We will break down the viewpoints of those who are in favor and those who are opposed. Proponents that argue the following reasons genetic modification of humans should be allowed. It can eliminate genetic diseases. By incorporating other organisms' DNA into our own, we are diversifying humankind. It can enhance beneficial traits such as overall health and compassion. It really isn't changing evolution, just accelerating it. It really isn't very different from cosmetic surgery or taking drugs when you are sick, which also alter natural biological processes. And finally, it is just another form of parenting, like sending your kids to an expensive college or paying for a tutor so that they do well.
This idea is explored more in the following clip.
We attempt to design our children, if you want to put it that way, when we educate them, when we try to instill social ideals, when we try to make them cooperative, social beings, when we take them to violin or sports lessons. All of these things attempt to shape our children into people who will have better lives. There is no reason to treat these environmental interventions any differently to direct biological intervention. What we want is our children to have better lives. How those better lives are brought about shouldn't make a significant moral difference.
Opponents of this technology argue that the expense leads to too many dangers because, on one hand, designer children will have an advantage, just like the animals that we talked about earlier. This also means that society will split into two genetic pools, one that encompasses the rich and another that encompasses everyone else. It's never going to be anything more than a plaything for the rich, so why invest precious research dollars into it? Also, because only the upper class could cure themselves of genetic disorders, these diseases could become a characteristic of the lower class only, and thus will no longer receive attention from researchers and innovators. Those against GM humans express concern that more babies will be chosen for their appearance than for being disease-free, essentially a corruption of this technology. More extreme opponents threaten that people will abuse the system and the technology to create superhumans who have a genetic tendency towards evil, or that some maniac will genetically engineer an army of people who can't feel pain. The last main argument of the opponents is that choosing sex, or any trait really, is inherently sexist or discriminatory. Many people feel our society needs to be 100% politically correct, and so these isms are horrid, antiquated beliefs that should be permanently squelched out.
In 20 to 30 years, we'll be able to control not just our personal genome, but the genetic heritage of the entire human race. So the question is, how far do we want to push this technology? I mean, already there's an international black market in human growth hormone as parents try to control the height of their kids. In the future, we'll be able to control the genes of our kids. Will we have a generation of designer children?
The three topics we covered were genetically modified foods, genetically modified animals, and genetically modified humans.
GM foods have the potential to increase total agricultural yields and reduce the need for sprayed pesticides and herbicides. But since research has not been done, nobody really knows what the consequences of inserting foreign DNA into plants might be.
GM animals are of interest for many of the same reasons as GM foods. Animals could be designed to convert food more efficiently into food for us. Additionally, we could grow high-value chemicals and natural materials in animals. But because GM animals have an advantage over natural species, it is very likely that escaped animals can and will send the natural species into extinction. Additionally, current methods of genetically modifying animals are not completely successful, meaning crippled animals are common.
Designer babies are incredibly controversial. On one hand, the technology could completely eliminate genetic diseases from the population. It could result in the amplification of beneficial traits, such as intelligence, compassion, and general health. On the other hand, it might result in highly intelligent test animals, such as the apes in Pierre Boulle's Planet of the Apes, and it might give some humans extreme advantages over others. It could also result in the overemphasis of superficial things like appearance instead of the deeper things like personality.
So what do you think? We encourage you to use and share the information here to participate in educated discussions about what the future should hold. Here are some questions to spark thought and add to debates.
We hope you're all still awake. It's a lot of information to take in. If you have any questions, please post them in the comments and we will do our best to answer or address any concerns. Thank you.